Hello everyone. Before starting the presentation, I want all of you to imagine yourself waking up on a fine morning to brush your teeth, but you drop the brush in the sink. You try to tie your shoe, but the laces slips out of your hand. You know something is wrong. You go to the doctor, but the doctor is as confused as you. He just label it as some lack of sleep and sends you back home, but tells you to keep an eye on it. But the symptoms only get worse. You go to the doctor again, and a series of tests reveals a devastating diagnosis. You have amyotrophic lateral sclerosis, or ALS. Patient with ALS ends up paralyzed from head to toe, sometimes left with just the slightest movement of eye. Uh, uh, we At the moment we don't have any cure of it. Thus, me, Nazir, a medical student from Pakistan, along with my friends Laiba Imran and Hanfa Wazir, did a research project by comparing the differentially expressed genes of ALS and to look for the potential drug candidates for drug repurposing strategies to find a cure of this menace.
Sorry for interruption, but the slides are not changing.
Uh, 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 okay, ALS or amyotrophic lateral sclerosis is a motor neurodegenerative disease that affects the motor neurons of brain and spinal cord. Basically, it has impacted millions with renowned names like Stephen Hawking and Lou Gehrig. It is of two types: sporadic or non-inherited, or familiar or inherited. Combiningly, they affect 2 to 3 people per 100,000 annually. It starts with some mild symptoms like muscle weakness and twitching and ends up in complete paralysis. Currently, there is no cure of it. However, the current treatment regimen only ensures a slower progression of the disease. We decided to study the genetics of the disease. As you know, that genes are the software of life. They express in certain ways to produce protein. But if you have some defected or mutated gene, the expression in this case would call differential expression, and as a result, a disease or malignant protein is formed. So what we do in precision medicine, we spot those mutations and target them to some certain drugs. Thus, it is called tailored medication. Our objectives were to document and define the differentially expressed genes, to perform the gene ontology, and to investigate the probable drug candidates for drug repurposing. This was the workflow that we followed, started with the genomic data hunt on Geo, then data pre-processing, DEG identification, gene ontology, network analysis, protein visualization, and drug repurposing finally. For genomic data hunt, we went to Geo, Gene Expression Omnibus, and the dataset by Dangan et al. Was included in our study. It was downloaded and was pre-processed to different bioconductors. DEGs were identified using Lima package. There were 7,031 and 7,044 DEGs for sporadic and familial ALS, which were filtered on the basis of their adjusted p-values and log of FC count. Still, we got highly specific and highly significant 78 familial and 132 sporadic genes. The outliers labeled in red and blue are basically the culprit behind ALS. These are the top 5 differentially expressed genes of both sporadic and familiar, with names like AQP1, AXIN1 for sporadic, CFD, M52 for familiar ALS. The next step was to do gene ontology. It was done on David. The top genes were studied on the basis of their cellular component, molecular function, and biological processes. The next step was to visualize them. It was done on Cytoscape using string extension. This is the network we got for our 132 sporadic genes, while this is the network we got for our 78 familial genes. The next step was to do protein visualization. It was done on PDB. The next step was drug repurposing. It was done on DGIDB. We got total 8 sporadic and 7 familial drug candidate for our disease. They were further studied on drug bank on the basis of their indication and contraindication. So this is the therapy that we propose. It includes all the drugs that targets majorly all the genetic drivers that are responsible for our ALS. So we conclude that by using this therapy, we can ensure a much better prognosis for the patient of ALS. Thus, our conclusion was there are ample of differentially expressed genes for sporadic and familial ALS and potential drugs for drug repurposing strategies. Thus, by studying the genetic landscape of this disease, we can ensure a much better we can ensure a much better prognosis in future. In future, we want to take this project to the lab and to perform various in vitro and in vivo experiments to validate the efficacy of our drugs. These are the references that we use. So this was all about my project. If you have any question, please